Good morning and welcome to this session on Research Talks. My name is Sudha Chakrapani. I'm an associate professor in the Department of Physiology and Biophysics, and I'm the director for the Cryo-Electron Microscopy Core. And I'm very excited to co-lead this session with Tara De Silva. And this session features 4 stellar talks, and we'll start off with our first speaker, Dr. Jonathan Haines. He is the Mary W. Sheldon, MD Professor of Genomic Sciences and the Chair of Population and Quantitative Health Sciences Department at Case Western Reserve University. A little bit of background on Dr. Haines. He received his BA degree in biology from Colby College, and then he went on to get his PhD in genetics and cell biology from University of Minnesota. Prior to his arrival here at Case, Dr. Haines held faculty appointments and leadership positions at the medical schools at Harvard and Vanderbilt. He is the founder and director of the Cleveland Institute for Computational Biology. It's a collaborative venture between Case Western Reserve University School of Medicine, UH, and the Cleveland Clinic. So Dr. Haines has several national services to his credit. Just naming a few, he serves on the NIH's National Advisory Council for Human Genome Research. He's on the scientific advisory panel for the NIH's National Human Genome Research Institute Genome Sequencing Program. He is on the board of directors for the National Organization for Rare Disorders, on the executive committee of the NIH's Alzheimer's Disease Sequencing Project, and he also serves on the editorial boards of several journals, namely Neurogenetics, Human Molecular Genetics, and Nature Genomic Medicine. His research covers all aspects of epidemiology, the study of the role of genes and the environment in human health and diseases. And today he'll tell us about genetic studies of dementia in the Amish. And please welcome Dr. Haines.
Well, I want to thank you for the opportunity to give you an idea of some of the research that we're doing relative to Alzheimer's disease. It's hard to follow Dr. Rosen's talk, but you know, we tend to be a bit stressed when we're at work, and it's really nice when we go to Holmes County, where the Amish, a lot of the Amish live, helps to de-stress us a bit. So I'm going to give you just a brief, most of you already know this, a brief introduction to Alzheimer's disease, and then why we would study the Amish, and then some of the results that we have. So Alzheimer's disease currently affects more than 5 million people in the United States, and as we've already seen, the number of affected individuals is going to greatly increase as the population ages, so we're worried that we're going to have upwards of 15 million individuals by the 2050. The prevalence of Alzheimer's disease doubles every 3 to 5 years and approaches perhaps even 50% by age 85. So it's a very, very significant problem. The vast majority of Alzheimer's disease occurs later in life, after the age of 60. There are some rare forms that occur earlier on. The clinical diagnosis is usually pretty accurate. If you go to the Cleveland Clinic or you go to UH, you can get a really good diagnosis. But the definitive diagnosis can only be done on autopsy. There are both genetic and environmental factors that we know increase and potentially decrease the risk of developing Alzheimer's disease. And as we've heard, the treatment options are very, very limited, and virtually every drug trial that's been out there has failed, and failed miserably. So we need to find some other ways of trying to understand what's going on in Alzheimer's disease, and see if we can't identify ultimately some treatments, and potentially effective treatments, and potentially preventions. So I'm just going to give you a bit of a history of the genetics of Alzheimer's disease, which is what we're specifically studying. There was a lot of work that was done starting in the mid-1980s to try to understand Alzheimer's disease and the genetics of Alzheimer's disease. And the first success was in 1991. The first mutation was found that caused rare early onset Alzheimer's disease. And then in 1993, there was a breakthrough in identifying the first and still the major gene for late onset, the most common form of Alzheimer's disease. That's the ApoE gene. I'll talk a bit more about that. And then in 1995, there were a couple of other rare early onset genes that were discovered. And then you see this big hole here. We tend to call that the dark years because there was a lot of work done to look at specific candidate genes to try to identify more. And the problem with that was we did not yet have the tools available to really search through the genome. Hence the Human Genome Project, which has been very, very successful and very helpful to us. So after all of that, we started to be able to search the entire genome for other genes that might affect Alzheimer's disease. And in 2009, we were able to publish the first genome-wide association study to really look at that, identified several loci and genes that were involved. And then since that time, we've been able to start to do whole exome and whole genome sequencing in samples and be able to identify rare variants that are involved in at least some cases. And much more recently, we've been able to do more whole genome types of sequencing to really look and identify other common and rare variants that are involved. So this gives you a sample of the most recent data that's out there that's published earlier this year, where we now have approximately 24 variants, loci, genes that are involved in late onset, the more common variety of Alzheimer's disease. And this was done collecting samples from all over the world, putting together a lot of different data sets. So nearly 100,000 individuals were used in this study. And I'm not going to go through all these details. This is what's known as a Manhattan plot. And all it really shows you is that's the whole genome from chromosome 1 to chromosome 22, and the number of the genes that are involved in that, the genes and loci. In some cases, while they've listed a gene for every one of these, it's not necessarily that gene. It's a variant that is somewhere close to those genes. So to summarize where we think we are at the moment, you can see this pie chart. ApoE still probably explains upwards of 20%, 20 to 25% of the genetics of Alzheimer's disease, late onset Alzheimer's disease. The additional loci, genes that have been identified using the genome-wide association studies, probably in total explain maybe as much as 18% of the genetics of Alzheimer's disease. So you can see that a lot of those loci actually have very small individual effects. The early onset genes maybe explain 1 to 2% of what's going on out there. There's some rare variants in the later onset that might explain as upwards of 5 to 10% in the end. But what we're really still struggling with is this large green area over here where at least half of the genetics of Alzheimer's disease still has not been explained. And we still need to try to find ways of identifying those. And as you can see, we've already looked at 100,000 individuals. And one possibility is to go and find another 100,000 or 200,000 individuals. And actually, in some cases, that's ongoing. But we actually can take a different approach to try to identify things. So I'm going to go back to ApoE for a minute just to be sure that everyone's on the same page. ApoE, apolipoprotein E, has 3 common variants to it. Polymorphisms. The ApoE2 allele, which is about 7% in the population. The ApoE3 allele, which is by far the most common, 75-76% allele frequency in the population. And the ApoE4 allele, about 17% in the population. The ApoE4 allele is the one you worry about because that's the one that carries a 3-fold increased risk of developing Alzheimer's disease if you have 1 copy. If you have 2 copies, it's actually closer to 10 to 12%, 10 to 12-fold increase in the risk. So if you're an ApoE44, your risk is really very, very high. The ApoE2 allele, on the other hand, is actually protective. So it actually cuts your risk in half if you happen to have that. Of course, it's the rarest of the 3, but it does, in fact, have a protective effect. And then the ApoE3 allele is the most common one, and that's the one that tends to be considered neutral for Alzheimer's disease risk. So remember, the ApoE4 allele is increased risk, 2 allele is decreased risk. So why would we study the Amish as a different way of trying to identify genes that are involved? So the Amish emigrated from Western Europe in the 1700s and the 1800s. There were 2 waves of emigration. They are a religious and cultural isolate. One of the reasons they emigrated from Europe was because they were persecuted there and not treated very well. Their lifestyle increases the homogeneity of their exposures to the environment. So their formal education is considered, more education is considered protective for Alzheimer's disease, but their formal education tends to stop at about the 8th grade. The good thing is there's virtually no alcohol or tobacco use, and they tend to marry within their faith. So because as part of their faith, birth control is not something that they consider, they tend to have very large families, particularly in the older generations, and that leads to a very large, remember they're an isolated population, relatively isolated population, so very large, very interrelated families. And that has a lot of advantages when you want to start looking at the genetics, because you can actually see how the variants and the polymorphisms and the diseases segregate through the families. So this is a genetically isolated founder population, and one of the nice things about that is if there is a rarer variant that has an effect on whatever disease you're looking at, in this case Alzheimer's disease, you're more likely to be able to see it if it's segregating in that family, because it's going to be much, much more common than it is in the general population. So rather than having to identify 300 or 400,000 individuals to find these rare variants, we might be able to do it with just a couple of thousand individuals. So there are approximately 250,000 Amish in the United States. The 3 largest concentrations of Amish are in Ohio, Pennsylvania, and Indiana. Ohio and Pennsylvania, it's interesting when you talk to the folks there. If you talk to the Amish in Pennsylvania, they say they are the largest. If you talk to the folks in Ohio, they say they are the largest. And in fact, it really, depending on who you talk to and what data you look at, it sort of goes back and forth. So basically, they're about the same size. Indiana's the next highest, not quite as large. Our source Amish populations are the Ohio and the Indiana Amish. So those are the groups that we're actually working with. So as I said, the Amish emigrated to the United States from sort of the Swiss, German, French area border. And there were 2 waves of emigration. The first wave in the 1700s went primarily to Pennsylvania. The second wave, which was in the early 1800s, some of those went to Pennsylvania, some of them went to Indiana and Ohio, and there was an out-migration from Pennsylvania to Ohio and Indiana. So there's been a bit of a mixing between those. It's very clear from the data that we have that the Ohio and the Indiana Amish are much more closely related to each other than they are to the Pennsylvania Amish. So one of the interesting things, and one of the aspects that we've had to work quite a bit about, was how do you actually ascertain? How do you actually get in with the Amish communities and be able to work with them? Now they are, as I said, culturally isolated. They live in very rural areas. They've got historically a very agrarian lifestyle. So we can't use the usual methods of ascertainment. We can't send out letters, we can't call them on the phone and do all of that kind of thing. So contact primarily is now door-to-door. Letters sort of work, but the door-to-door is really what works the best. So we're just going down the street, knocking on doors. And you say, well, you know, if they were living in Cleveland, you'd knock on the door, no one's going to answer, right? But in the Amish communities, they're very, very open to this. So that's the way they communicate. That's the way they get in touch with each other. So it actually works quite well. We did spend probably the first 2 to 3 years of our research, 20 years ago now, actually just getting to know the Amish communities, having them know what we were about, and that we were going to be around for a while doing these studies. And once they became comfortable with us and we became more aware of their lifestyle, it's been a fantastic interaction. They've been very, very open, very cooperative. They're very altruistic. So we've been able so far to ascertain probably about a third of the individuals over the age of, say, 60 or 65 that live in these communities. One of the ways that we've been able to do this and do this effectively is that our ascertainment personnel, the ones that are out there knocking on the doors, actually live in those areas. They live in Indiana, northern Indiana. They live in central Ohio. They've grown up with the Amish communities. They understand it very well, and that works really, really well. So because we've had such a good relationship, we get participation rates of approximately 90%. Sort of depends, but approximately 90%. So virtually everybody that we talk to is interested and willing to participate if they can participate. So the first question 20 years ago that we had after we published the APOE findings was, well, you know, is there actually Alzheimer's disease in the Amish? Now, if you were paying attention to Dr. Rosen's talk earlier, he had a list of 6 things that you should do to reduce your risk of Alzheimer's disease. The Amish actually do 5 of those things already. Okay, the one thing that you could argue about is their diet, because it's a very farmer's diet, very heavy in dairy and meat and potatoes and that kind of thing. But everything else they're basically doing already. So there's a question about that. So there was an initial study done that suggested that dementia in general was a little bit lower in the Amish communities than it was in the general population, but that what dementia there was seemed to congregate and collect within certain SIBSHIPs and certain subparts of the pedigrees, suggesting that maybe genetics was important. We also took a quick look at APOE and in one of the small isolates, and did not find any APOE4 at all. And in that case, so if they're having Alzheimer's disease, they're already reducing their risk by their lifestyle, and yet there is Alzheimer's and it's congregating family, suggests that there's other genetics involved, which is why we decided that we should look at it in more detail. So the first thing we did was to go back and look at APOE in a much broader population of Amish, and we in fact found that they have the 2, 3, and 4 alleles. You'll note, however, that the APOE4 allele is somewhat less frequent in the Amish population. The 3 allele is more frequent in the Amish population. But for individuals who have the APOE4 allele in particular, we see the same kind of effect. They have an earlier onset and a higher risk than individuals who don't. So we're seeing the same effect of APOE when it's there, but it's still not there at the frequency that it is in the general population. So APOE is not explaining everything. So the next question is the genetic architecture of Alzheimer's disease in the Amish the same as it is in the general European Caucasian population? So I go back to that Manhattan plot that I showed you earlier, and what we did was to generate a genetic risk score that included, we actually did this a little bit before this paper came out, so we used most of those loci and did a genetic risk score which basically sums up the effect of all of those genes and loci that are involved in Alzheimer's disease, and we can create a score out of that. And then we can calculate that score for every individual and then see whether or not those scores on average are the same, higher or lower than they are in the general population. And what this graph shows is that if you look at the Amish cases and their average score and you compare it to the unrelated Caucasian population scores, they're much, much lower, which suggests that in fact they have a different architecture and that the genes that we know about are not what's explaining a lot of the Alzheimer's disease in the Amish. You can see that it's somewhat higher than the Amish control, so there is some contribution there, but it's not as high as it is in the general population. So something else is going on. So that was really encouraging. So about now 7 years ago, we did an initial genome-wide association study to see if there were novel loci. It looks like there should be novel loci out there. So are there? And so we took a look. And this is a different way of coloring a Manhattan plot. But we found one suggestive, it was not genome-wide significant, but one suggestive locus that might be involved. But this would require a lot of follow-up studies to understand that. Now, I'm going to show you, this is a part of what the actual pedigree for all those people looks like. We have over 5,000 individuals. If you go back 13 generations, we can connect them all up. And so you can see, these are the generations, these are all the people and all the kind of things. So this is the kind of thing we have to deal with when we're trying to do our genetic studies. And so when we're trying to look at how the different variants actually travel through the pedigree over the generations, it obviously becomes computationally impossible to do it as one large pedigree. So what we've had to do is to split it into a whole bunch of smaller pedigrees to try to get the information, actually allow the computers not to overheat and not to spend 5 years calculating. And when we did that, we actually identified 4 regions across the genome that look like they probably have some variant in them. And they're on chromosomes 2, 3, 9, and 18. And just to orient you here, a LOD score of greater than 3 is considered pretty good. We've got scores of 5 and 6 and things like that. So this is looking pretty good that there's something there. The problem with this technique is that it doesn't give us the variant itself. It tells us that there's 2 or 3 million, maybe 15 million base pairs that we have to look at. Okay, so there's still a lot to look at. It's not the whole genome, it's not 3 billion, but it's still 15 million variants that we have to look at. And that's not so easy, especially back in 2012, 2013. So the status of where we were about 6 years ago was that we had enrolled about 2,800 individuals. We had DNA samples that we'd collected on about 1,100 of those individuals. We had 162 with dementia, likely Alzheimer's disease. We had these strongly suggestive results, but then where do we go with that? Our problem was that the methods for association, accounting for that complex degree, the complex pedigree, not so good. The linkage analyses, what I just showed you, are difficult and complex pedigrees, and we still have the searching through the 15 million base pairs to go through. And the SNP array data is incapable of identifying the actual rare variants. So what we've done now is to reimagine our study a little bit, to take advantage of the advances in the computational, the genomic, and the statistical methods that are out there. So we designed a new study based on all the information that we had that allows us to leverage all the unique aspects of the Amish. We're doing the ascertainment in Ohio and Indiana. We're going to identify affected individuals with Alzheimer's disease. This is sort of the classic approach. You're going to identify the affected individuals. You're going to try to find additional risk loci variants that are involved. We're also going to identify cognitively normal individuals who are at high risk of developing Alzheimer's disease but haven't yet developed Alzheimer's disease. Those individuals, our hypothesis is, have protective loci, something that's actually protecting them from developing disease. And we'd really like to know that because maybe we could use that information to help develop the treatments or the preventions more broadly. We also intend to ascertain all the individuals over the age of 80 across these, and that allows us to do more epidemiologic studies, start looking at environmental and lifestyle factors and things like that. So we're doing cognitive screening on all these individuals. We're going to collect blood and plasma samples, and we're going to revisit the cognitively normal individuals every 2 years to see if they maintain their cognitive status or they start to decline. So when we say we're looking for cognitively normal individuals who are at high risk, what do we mean? What we're doing there, the criteria, is that they have to be cognitively normal in all our neuropsychological testing. age above or at age 80, and they have to be related to somebody who had Alzheimer's disease below the age of 80, so a sibling perhaps, who had, or a parent who had Alzheimer's disease, or that genetic risk score that I told you. If they have a high genetic risk score and are still cognitively normal, that also suggests that something is preventing them from developing disease. And these just give you an example of some of the pedigrees that we have. Those are actually the ages in there, if you can read them. So we have a number of individuals over the age of 90 who are actually doing quite well. So as of right now, this is where we are. The project's been ongoing for a couple of years now. We've enrolled over 500 individuals so far. Just to give you an idea of how difficult this is and how much time and effort actually goes into ascertaining these individuals, we've gone back and we've identified with all these individuals we thought were out there, and it turns out that the majority of them are now deceased, unfortunately. We've identified a number of individuals who have declined to participate because they are already demented and they're so far down the road that they're not willing or able to participate in the study. And our rate of decline is only about 7%. So if you eliminate some of those other things, the rate of decline is actually small. I also have on here the number of contacts that we've attempted. Remember, we're going door to door on this, so it turns out that a lot of the time there's nobody home, and because they're out in the farm, or they're going off to a wedding somewhere, or there's wedding season. Ascertainment goes down during wedding season because they're all off attending the weddings. So we've actually done a lot of ascertainment there. We have a case conference that typically is done about every month to 2 months to adjudicate all the results and identify individuals that we truly think are cognitively normal, truly think we have dementia. So far, we now have over 200 individuals who are cognitively normal. We've identified about 82 individuals that we've tested who are cognitively impaired in one way or another, and a small percentage of those probably actually do have dementia. And we have a number where we're going to have to go back because the data is unclear. So our next steps, what we are currently doing is we're generating these high-density SNP polymorphism arrays on 1,600 individuals. We already have such data on 700 individuals, so we'll have over 2,200 or 2,300 individuals that we can now study and do very detailed association studies. The methodologies have gotten much better and faster, so we can actually do that. And we can do that both for Alzheimer's disease and for cognitive preservation. We can do the linkage analysis. Those methods have gotten better as well. So we can do those kinds of things. We are also in the process, and those data are actually being generated right now. We should have them in the next couple of months. So next year we'll actually have results, more recent results to be able to show. We're also going to get whole genome sequence as part of the Alzheimer's disease sequencing project, the national project. We're going to get whole genome sequence on approximately 1,100 of these individuals. If the cost would go down a little bit more, we could do some more individuals. That will allow us to really do what we really, really want to do, which is to identify segregating rare variants that might have very strong effects, but in a small number of people. And we want to be able to do that particularly for the cognitively preserved individuals. We can also do it for those individuals who are declining. And because we're going to go back every couple of years, we'll be able to look at progression in much more detailed fashion than has been done in most studies, going from cognitively normal to mild cognitive impairment, MCI, ultimately to Alzheimer's disease. So we're hoping that we'll be able to continue this study for a number of years. So with that, I would like to acknowledge, obviously, this is a huge amount of effort from a huge number of people. I've got a lot of people in my lab and individuals who live in Holmes County who are doing the ascertainment and doing the analysis and doing the collection of the samples. I have a number of former students who have been working on this project who have now gone on to bigger and better things. I have Alan and Paula at UH who have been very, very helpful in doing a lot of the clinical assessments. A lot of the building that pedigree stuff is a group at NIH that's been doing that. And then our collaborators who handle all the Indiana ascertainment who are currently at the University of Miami. So with that, I'll stop there and I'll take a few questions if we have time. Thank you.
Thank you for the very nice talk. I have a question about the incidence of Alzheimer's disease in the Amish population. Is that less than in the general population? And do Amish people in general, do they have a longer lifespan? So is their age at onset of dementia sort of delayed than the overall population?
So the answer to the first part of that is we don't really know because a formal study of incidence hasn't been done in a long time. One of the things that we'd like to be able to do if we're going to go and look at everybody over the age of 80 is to actually see what that is. We can look at the prevalence, obviously, and then over time we can look at the incidence. All the data that we have so far, and certainly our experience, suggests that it's about the same as in the general population. Their lifespan is about the same as well. So we're not seeing a whole lot of difference between that and the general European population. Yeah. Yeah.
So right now you're doing this with an eye towards Alzheimer's disease. I'm wondering if it is possible with the participation of these people to look at maybe one or two other factors that impact other neurodegenerative disorders?
Such as?
Such as, I mean, if you're looking at, I don't know if the incidence of Parkinson's disease is particularly of interest.
So it is. I actually had, I don't know, this must have been 20 years ago, I got a grant from the Michael J. Fox Foundation to actually look at Parkinson's in the Amish. We had identified about 30 individuals that looked like there was a nice pedigree there. We did a lot of the studies. We couldn't actually find anything in particular there. One of the issues with Parkinson's that we didn't anticipate is that they use a lot of pesticides. So anyone who thinks that the Amish only grow organic food, they don't. They use a lot of pesticides, and they're not very careful about their exposures. So there's a lot of issues there. But there is more Parkinson's in the Amish. We just have not, I mean, we've studied in the past, but we haven't studied it recently. Any other questions?
It is known that the Amish populations have the incidence of genetic anomalies like hair lip, club feet. Is there, just for example, is there any correlation that you're looking at between these other effects and then your data?
There is no correlation that we know of at this time. So almost all of those rare diseases that are showing up more rare in the general population, showing up more commonly in the Amish, almost all of those are childhood onset kinds of things or things that are chronic and go forward. We've not seen a connection with those. Once we have all of that genetic data, we can actually look directly rather than just by report. So that's something that we will be looking at in the next couple of years.
No further questions. Next time, Dr. Haines again.
All right, thank you.